Hello everybody, welcome back to Cells and Tissues, the biology of cells and tissues. This is still going to be the first week's chunk of material. We're talking about these cell junctions and surface modifications. So we're going to go over defining the cell junctions, talking about which junctions are on the lateral versus the basal surfaces of the cell. 5 main types of cell junctions, various sites of their distribution, components of the junctional complex, as well as the structure and function of some of those surface specialized structures. Looking at this in general, right? So when we talk about epithelial tissue is going to be the kind of tissue that is going to coat a lot of our organs. Think the intestines, think the bladder, think like a lot of things that we require that are going to have absorption type qualities. Okay. Little vocab, apical surface. That is the surface that is facing towards the opening. So like if we're looking about the intestines, this would be the apical surface is what is going to the empty space in the middle of the intestine, or where like the food and stuff is going to be passing through. Lateral surface is where the epithelial cells are connected side to side. Basal surface is on the bottom, and they connect to a basal membrane made up of a basal lamina, and then a little bit deeper, a reticular lamina. So kind of like if we were looking at, let's say we would take a part of the intestines, the long part of the intestines, we cut it and look straight down into it, and we see the intestine right there. We are going to have our, the deeper layer is going to be our reticular lamina. Then we'll have our basal lamina, and then we have our epithelial cells inside of that, and that's the empty space, where stuff is passing through. Okay, so talking about a cell junction. So cell junctions, these are specialized attachments between adjacent cells. So this is connecting the cells to other cells and to their origin point. This is how the cells stay where they're supposed to be. We talk about, the body being 70% water, right? There's a whole bunch of stuff. If we think if there wasn't something to be holding all of this stuff together, holding all of our cells together, we just kind of melt into this pool of jelly. So that is the purpose of cell junctions. They are modifications of the plasma membrane. Remember that phospholipid bilayer that's all around all of our cells. The cell junctions are modifications of that. They provide the anchoring of adjacent cells to one another, and they also provide cytoplasmic continuity between cells. We'll look at how cells are connected to one another, how they interact with one another, how they're able to send signals to one another. Okay. Because as we know, if all of our cells want to be connected, be on the same page, doing what they're supposed to be doing. The first one that we're going to talk about are tight junctions. So tight junctions, their function are to seal the cell-lined compartments. Think we have 2 cells right next to each other. We have the little bit of space in between them. Tight junctions are like a needle and thread sewing these tooth parts together so that stuff can't go in between the epithelial cells. Okay? So like I said, they prevent water-soluble molecules from passing through the extracellular matrix. So the extracellular matrix is kind of that fluid, that is fluid slash stuff that is outside of the cells. So it stops that from going between the cells. It fuses the plasma membranes together. Protein strands and adjacent membranes fuse them. Like that's kind of like the thread that I was talking about, those protein strands. Maintains the polarity, so like the electrical charge of the cell, restricting membrane proteins to either to the apical, the top, or the baso, bottom, or lateral surfaces of the cells. Big thing to understand here, this is controlled by calcium concentration. this is a question that they like to ask on tests. That the tight junctions are controlled by calcium. Even more simplified, as you see, with these lectures, I've been trying to go into even greater detail. I've got a lot of extra information that's all down in here. Most of this information is not necessary. Focus on what is on the slides themselves. But I've tried to add a little bit of extra information, add extra context, and if you can go and you want to read through the things, you're going to learn a lot more with all of that extra information. So, the tight junction, again, those are specialized structures, form a barrier between the neighboring cells, prevent leakage. Now, these are formed by a network of proteins, specifically 3 main proteins. These are called claudins. I'm actually going to highlight these. Claudins, occludins, and junctional adhesion molecules, or JAMs. Okay, claudins and occludins are the ones that they focus on for this course, but you may see JAMs come up every now and then. This is a little bit confusing. Confirm with the professor, whatever professor you have, how they want to go about this. Most commonly, they say that it is the apical lateral surface, but technically there are tight junctions on all different surfaces of the cells. So check with your professor specifically. What do they say? Do they want you to know apical surface, lateral surface, basal surface, or a combination of those? Kind of the research and, as you dig deeper into biology, you can technically find them in all 3 of those locations. But for this course, depending on your professor, will depend on kind of what they are asking. Here's just some pictures of those tight junctions. You can see kind of that thread, those claudins and occludins making up the tight junction, really just kind of sewing it together and holding it together. These pictures that this professor has provided looks a lot more like that it's on the lateral surface. So if I were to lean towards anything, I would say lateral surface. As well, so these cell membranes are basically, again, these fused together. The big places that we focus on where these are is the places that we don't want leaks to occur. So the blood-brain barrier is keeping fluid and everything in the brain where it's supposed to be. Not too much fluid, not too little fluid, all that kind of stuff, keeping things out that aren't supposed to be there. That could mess with the brain chemistry. As well as the bladder. We don't want the urine leaking out. So specifically those 2 points are the specific spots where it's mentioned that the tight junctions are. More pictures, again, of those tight junctions. Just a reminder about that calcium. Moving on, Adherens junctions, also known as the zonula adherens. So this is a membrane protein, a calcium-dependent cadherin. This is another one. Again, remember, we need to make sure that we remember that it's calcium dependent. It is an actin microfilament, so a very small string, in the cell cortex that anchors a cadherin strand membrane, glycoproteins of each cell that interact in the presence of calcium to strengthen the tissue. Now, in English. So the Adherens junctions are specialized structures that are going to they provide a really strong mechanical attachment between the neighboring cells. Just like the tight junctions, although the tight junctions do join the cells to create a barrier, the actual adhesion of the cells together is not super strong, or not as strong in the tight junctions as it is in the Adherens junctions, aka the zonula adherens. They are made up of several different components. We have the cadherins, we have the catenins, and we have also part of the actin cytoskeleton. You can pause, and if you have my notes, again, those are down below in the description. You can go on to the link in order to get all of these. It goes into just a little bit more detail, as well as down here, a little bit more detail about the Adherens junctions if you want to know more or just still don't quite understand what's going on. Another little bit of vocabulary. when we're talking about these adherens junctions, where all of these junctions, these adherens meet with like the actin filaments, everything where they all meet, we call this the adhesion belt. So you have a whole bunch of these right here, of these proteins all working together, all of these cadherens all coming together kind of at one point, just below the tight junction. Most commonly is what the current literature says. So just underneath that, again on the lateral surface, we have this thing called the adhesion belt, where that zone is of all of these adherens. Again, here we can see some of the other things. We can see, like we talked about, they have the actin filaments, that they have catenin, that they have the cadherin. I have a couple extra tidbits in here. I threw in some definitions, as well as a bunch more information on the actin skeleton down below, if you want to go and look through all of that. Now, desmosomes, the next one. So we have the first chunk, adherens junctions. Now we talk about desmosomes. So these are going to be similar in function to zonula adherens. Okay, they have the same job. Biggest differences, these are disc-shaped. That's an easy way to ask these type of test questions. They'll throw that as a keyword. They'll ask something like this disc-shaped protein, or this disc-shaped junction, or this disc-shaped whatever, does this job. And it's going to describe almost identically what adherens junctions does, except that disc-shaped is that keyword for us to know that we're talking about desmosomes. It's a cytoplasmic intermediate. Cytoplasmic intermediate filaments anchor the cadherin membrane proteins, which are called desmoglein and desmocolin, that bind the cells together. Cytoplasmic intermediate filaments, it's like string-like structures. They're just extending. We can look at them here. They're extending out from the desmosomes into the cytoplasm, which is why they're cytoplasmic intermediate filaments. They are filaments going into the cytoplasm. Intermediate meaning they're attaching or meeting each other on different sides. And again, its whole purpose is to strengthen the tissue. In here, we can see those types of cadherens, those desmocolins and desmogleins. They're all these cytoplasmic intermediate filaments are also known as tonofilaments. These are keratin intermediate filaments. I don't recall, it was when I took this course, that they ever mentioned that they were keratin. But in case, that does, I don't recall that, but it doesn't mean that it wasn't there. So in case it is there, you know that the tonofilaments are made up of keratin, which is common. One of a very common thing that we get actually from our skin. That is our desmosomes. Now, hemidesmosomes. Anytime that you see the word hemi almost always means like half. Which, if you think of it, you look at it, it's like, okay, hemidesmosome, more than likely, this is probably going to be disc-shaped as well, but it's going to be, kind of half of a desmosome. That's what we're going to be looking at. This is our guy that is going to be in the basal lamina, so down below. The other ones that we've looked at so far have all either been like the tight junctions. We'd call those apical/lateral, leaning more towards the lateral side. The zonula adherens, we call that lateral. Desmosomes we call lateral. And now the hemidesmosomes are on the basal lamina. Again, lateral surface, apical surface, basal surface. That basal lamina, connecting the bottom to the basal lamina. Here is what they look like. Again, the little half disc, whereas instead with the desmosomes, we have the two discs. We have the one single disc. From the cytosol, we have, again, those same filaments coming up through. The biggest difference here when comparing desmosomes to hemidesmosomes, and actually I know I have it written down below, but I'm going to add it in to here as well. Is that instead of using cadherins like everything else does, this uses what are called, make sure I spell it right, matrix integrins. Matrix integrin, another type of protein. There we go. I want to just highlight matrix integrins. There we go. There's matrix integrins attaching, again, the bottom of the cell to the basal layer. Okay, moving on a little, we're going to switch gears a little bit. So all of these were for, sticking stuff together. Now we're actually going to talk about cell communication. Cell-to-cell communication. And that is through something called gap junctions. So gap junctions are something, this is something we're going to talk about quite a bit. We're going to touch on it here in this course. And then we're going to get more in depth about gap junctions and kind of why they're important and what their role is when we get into Physiology 1, when we talk about cardiac, we talk about smooth muscle, we talk about all those kind of stuff, and how these gap junctions relate to what are called action potentials in order to actually cause muscles and stuff to move and to contract. Okay? It's a little bit ahead of our schedule right now, but we're going to see that later on. So, gap junctions are also known as communicating junctions or connecting junctions. Think of it like proteins that make up a tunnel to let stuff pass through. Important in cardiac and smooth muscle, allowing these cells to signal each other, to talk to each other, and to quickly pass on a signal or information to one another so that they all stay on the same page and are all acting consistently. You can see right here, it mentions that the cells are connected by connexins. Connexins are the proteins that make up the gap junctions, and they have 6 subunits. Easy test question right there, that they can ask how many subunits are there for a gap junction. 6. There are 6 connexin units that all put together form a gap junction. I'm not entirely sure if technically you consider it 12 instead of 6, because you have 6 on this side, 6 on the other plasma membrane, and then they meet in the middle. But for the purpose of the slides and test questions that you'll get, as long as it says 6, the test question, quiz questions, exam questions that you will get are going to be that there are 6 connexin proteins. Okay, and again, we'll come back to gap junctions in future courses. Just a little note of medical application. Certain proteins of Zonula occludens there it is, the tight junctions. Zonula occludens. I'm going to put that in there. Tight junctions provide targets for some bacteria which cause food poisoning. And blistering disease involving the epidermis are due to abnormal desmosome function. Blistering diseases related to desmosomes. Tight junctions can be related to food poisoning, so make those kind of connections. This little chart, a lot of this chart is just review a lot of, what is up above before. Mainly focus on these top 2 columns as far as exams, understanding what components are in each of the different types of junctions. But read through all of it as well, to look, and it's additional good information. Terminal bar. One sec. Arial. I don't know why that font changed. I like consistency through everything, so let's make sure we change that. Okay. Okay, so a terminal bar. So a terminal bar is a group of junctional complexes. So we've been talking about different junctions. And so when we kind of lump them all together, we group them in a specific location called a terminal bar. So the tight junctions, the zonular adherens, and the desmosomes, we get this area when we look at it with a microscope. We can look at stuff using microscopes. You'll learn more about this in microbiology, but there are different types of microscopes. The most common and kind of the cheapest and the one we've used the longest is a light microscopy. Basically, you shine light on something, you have a lens, and it zooms in real close, and you can see stuff that's really, really small. With light microscopy, detail isn't always the greatest, especially with really, really small structures, and that's what we have here. So using just light microscopy, it just appears as a singular bar towards the apical surface of the cell, where these structures, the tight junctions, zonular adherens, and desmosomes, cannot be resolved. Resolved is just a fancy way of saying seen. They can't be differentiated. They can't be separated. But there's another type of microscope called an electron microscope. This literally uses electrons. It shoots little electrons into everything, and it has a reader that reads the electrons and the way that they interact. And this you can see a lot deeper and much more detail. So here we can actually see each of these different zones on an electron microscopy. Now, again, switching gears to surface structure specialization. We're going to talk about three things: microvilli, stereocilia. Should be stereo. Let me double check that that's spelled correctly. Yeah, it should be stereocilia. Okay. Microvilli, stereocilia, and cilia. Okay. All of these have similar type jobs. They do it slightly differently, have slightly different methods and characteristics to them. Overall, the main functions is to increase surface area that is available for interaction. So if we just have, here's the top of the cells and we have a straight line, there's not a lot of surface area here. But if instead we have all of these little projections here, all the way across, right? A whole bunch of them. Now, surface area-wise, we have all the way up, all the way down on every single one of these, and we greatly increase the amount of surface area that we have for interactions between ions, between chemicals, between whatever it is that we need to be coming into contact with, to interact with. Starting with microvilli. Microvilli are about 1 micrometer long. That's the symbol for micrometer, also known as microns. It all means the same thing. They are covered with a sugar residue that also has membrane lipids and proteins called glycocalyx. This is a very, very good test question right there. What is it? Which of the following has glycocalyx? Microvilli do. They form a brush or a striated border. Again, more keywords that we can easily be tested on. They are found extensively on the apical surfaces of absorptive cells. So we think absorptive cells, big ones. Think about the intestines. Microvilli are big into the intestines. Again, I have a bunch more information down here. So small intestines, kidneys, other places like placenta, gallbladder, those kind of areas is where you're going to find the microvilli, especially small intestines. Other important things, they have a core of actin filaments arranged longitudinally, so lengthwise, and these are not motile, meaning they don't move on their own. They just kind of sit there and they sway all on their own. Here we have the filaments, the actin filaments on the inside, which make them up. You have the microvilli, all of this stuff, and this cell coating. This is what these microvilli look like. These are very, very small. Microvilli, actin filaments in the intestines. You can see this is a drawing of how these are put together. There's the glycocalyx, that cell coating. You can see it's built. It's like this branched chain of stuff. It's got, it's got the sugars, it's got lipids, it's got proteins all built into it as part of it. The terminal web. So the terminal web is located on the cell cortex, made up of actin filaments, which is why it's called a web. This is where all of the actin intercrosses and stuff, and you can see this actin runs into the terminal web, keep everything anchored, keep everything in place. There's a little cross section. You can see the little microfilaments on the inside. We'll compare microvilli to each of the other parts, especially to cilia, because there are some key important differences that we want to look at. Stereocilia essentially are the same thing as the microvilli, except these have they are long and they are branched. Increased surface area for transport across the membrane. So you can see they look almost exactly like microvilli, just they are a lot longer. So that's really the only difference. So if we get test questions, make sure you know microvilli are 1 micron or less long. Stereocilia are much longer and they're branched. See, these are kind of connected. They're branched. Long. Cilia. This is one where a couple more details come important. So cilia, these are between 5 and 10 micrometers long, so 5 to 10 times as long as the microvilli. And then we also group flagella into them. Flagella are the longer version of cilia. Flagella we don't talk a lot about in this course. You'll talk more about flagella in microbiology in Try 2, because a lot of our bacteria guys, they have flagella that they use for moving around. So we'll talk about that more in that course. So these are motile, which means that they move. They're also known as axonemes. So if you see cilia or axoneme, it's the same exact word. The structure, the way that they're built up, is kind of two different parts. So at the very, very base, we have the basal body. So if we were to zoom really, really deep into the cilia, the very bottom, it has the basal body, also called a centriole, and there are 9 triplets of microtubules, so 18 in total. So 18 total microtubules, right? Because 9 times 2 is 18. As we go up towards out into the cilia, we actually gain additional central microtubules. We added an additional 2, right? So we have the 9 doubles, an additional 2 in the middle. So depending on how the question is worded and the answer choices that you have, 9 doublets of microtubules gives you 18, plus an additional 2, you have 20 total microtubules. So depending on the way that the question is asked, just kind of read it carefully. Does it include the doublets? Does it not include doublets? What is it actually asking, and what are the answer choices available to you? Again, sometimes teachers and everybody are human, so they might make a mistake. And as long as you can support your answer properly, a lot of times you can talk to the professor and say, well, the question wasn't clear because it didn't mention the doublets or not, so that's why I picked this answers. So if you know your stuff, you can usually talk to the teacher and get points back on tests as long as you know what you're talking about and the question if it was not worded appropriately. Big thing to remember, the major protein that makes up the cilia are the axonal dynein. And because it is motile, it needs energy, so it requires ATP. We mentioned that ATP is our energy source. And it has some supporting proteins, and we already talked about the basal body. A bunch more information down below about the cilia if you want to check that out. Looking at the pictures, there's those dynein arms. They have those microtubules. The dynein arms are kind of branching off of the microtubules to keep things connected, to keep things all together. ATP is used to help these move. And yeah. Last little bit, kind of comparison. I went ahead and put this chart together that shows the differences between the cilia and the microvilli. Again, the microvilli stereocilia are just long microvilli, so a lot of this is similar, except for things like the size and that kind of stuff. Differences that we want to remember, like size, movement, function, the primary function, that core structure, location, and then also that glycocalyx. I have all those highlighted. The big ones, important ones. We've talked and mentioned all of these in this recording, so none of this should be new information. This is just putting it all together here at the end. Okay, thank you guys so much for watching. Hopefully this helps a little bit understand better these cell junctions and everything that goes on with them. Have a wonderful day and good luck with your studying.